So guys, welcome to part 3. So far we understand quite a lot of basic things. So let's take let's make a diagram of a human cell. The way I used to draw this back in biochemistry. This is the bilipid layer, guys, ne? Bilipid layer. This is the bilipid layer. This is the cell of a human body, the cell membrane, and then inside there we've got the nucleus that has got nuclear pores. The nucleus, right? Yeah, and we've got the other, that's the cytosol, we've got the other endoplasmic reticulum, we've got the genetic material there, and and your mitochondria, that type of a thing. This is the human cell. And then on the other side, we've got the virus, which also has got this as well. Bilipid layer. So the bilipid layer that we have is not different from the bilipid layer that the virus has, right? Inside here we've got the capsid, and that capsid has got 4 things in it. That's the virus, right? It has got 4 things in it. The first thing that it has got, it's not 4, it's 5. It has got 2 strands of the viral RNA. It has got 3 enzymes. So the reverse transcriptase, the protease, and integrase. Acronym RIP. So those are the 5 things that are inside this. And that's the capsid. That's the matrix. And that's the bilipid layer. So guys, like we discussed on the other side, that this lymphocyte is the one that we spoke about in the other video. It has got okay, here we've got the GP41, glycoprotein 41, and we've got the GP120. These are receptors, right? That's the virus. And then on this lymphocyte, like we said, that we have that CD4 molecule. And we said that on the previous video that the CD4 molecule, as much as its job was meant to enhance or to help with the interaction between the MHC and the TCR to fight off infections, the virus uses that CD4 molecule to come and attach to this cell. So if I were to draw it more simpler, you've got now this lymphocyte with its genetic material there on the nucleus, with its bilipid layer, and with its CD4 receptor molecule. Now it has come close to the virus, which has got the GP120 and the GP41. Now when these two come together, there is another co-receptor that gets activated, the CCR5 and the CCXR4. I hope I get this right. If I make a mistake, I'll just type on the comments. But the most common is the CCR5, chemokine receptor 5. So the interaction of the CD4 receptor with, so this is what makes lymphocytes vulnerable to the HIV virus. It's because that GP120, which is a receptor or a molecule in the virus or on the surface of the virus, is the one that has got the ability to attach to this CD4 molecule. So which means that a cell that does not have a CD4 receptor on its surface cannot really attach to the virus. So once that happens, once this attachment occurs, these two cells are brought together. They are brought close together. Now you've got a situation like this. They are very close to each other now. Remember, this and this are the same. They are both bilipid layers. When that happens, there is an opening that occurs here, and this sort of like becomes one. So you've got to see something like this now. And then all that genetic viral material that is there, it gets injected into the lymphocyte. Now you find it here like that. Once the capsid enters the cytoplasm or the cytosol of the lymphocyte, it gets degraded, it gets broken down. Now you release all those 5 things that are there, you release them now into the cell. Now you have a situation like this. You have one cell now that is like this. You've got your nucleus there, with your genetic material. You've got your other organelles, and then now you've got this viral RNA that is in the cytosol with those 3 enzymes that came with the virus. This is what you have now. And the moment this virus does this and inject the lymphocyte with its genetic material, that this is the part where now lifelong infection starts. So it marks the start of lifelong infection. So in case you're wondering how are we moving, so we had this, we moved into this, and then we moved to here, and then now we are here. Now you've got a lymphocyte that has got the genetic material of the patient, and you've got a cell that's got the genetic material of the virus together, right? I'm gonna explain this again. So guys, I'm saying you have a lymphocyte, which is a human cell, and then you've got the virus. There are similarities between these. The bilipid layer is similar for these two. That's why it's easy for them to fuse like that there. They call it endocytosis. That's why it's because the material is the same bilipid layer. So what happens is that on the lymphocyte, you've got CD4 receptors which interact with the GP 120 of the virus, and also the GP 41 of the virus. So that interaction between those molecules brings these two together. It draws these two together to such a point whereby, like I said, the co-receptor there's also a co-receptor, the CCR5, and the main receptor there is the GP, the molecules, the GP 120 and the GP 41. So once that interaction occurs, you get a situation where there is endocytosis. So the material in this, which is the 2 strands of RNA and those 3 enzymes, the reverse transcriptase, the protease, and the integrase. So those 3 enzymes and those 2 single strand RNA are injected into the lymphocyte, and this becomes one cell, basically. Now you've got a lymphocyte that has been infected with the HIV virus. So now you've got 2 genetic makeups here, or 2 genetic material, one from the patient and one from the virus, and together with those 3 enzymes. Now let's talk about what happens when this has occurred. So this is our lymphocyte with our nucleus, with our genetic material, chromosomes, and other cell organelles. And right there on the cytoplasm, we have those 2 RNA strands from the virus, and we have got